All right. So hopefully some of the stuff you've already learned, hopefully that'll disappear in a moment, in your biology class in high school. But most of the stuff that you learned in this particular class are topics that you would have come across or should have come across in high school. See if I get this out of the way a little bit. So we talked about biochemistry already, or at least you did some of the notes. And I don't know how much you got through that. I don't know if you remember things like what are the building blocks of proteins? What do we call those? Amino acids. Good. What's the building blocks of carbohydrates? Complex carbohydrates. What are those? Yeah, very good. Disaccharides and polysaccharides is a complex carbohydrate. What's the building block of DNA? Yeah, nucleotides and nucleic acids. Good. So those are some of the things that were covered in the previous lectures. If you haven't seen that, I can provide a link, or you can look at Dr. Hum's notes for those. Now, again, we don't, the only planet that we know about life for sure is what planet? Earth, right? And we know that Earth life is composed of cells. And that's the only planet we know of. And so this was something that took a while for people to figure out because they can't see cells for the most part. They're microscopic. Now, there are some cells that are a little larger you can see, but anyway, it took a while for us to figure that out. We didn't have the technology, the tools like a microscope. So some of this original idea started to come together due to some early scientists like Robert Hooke, who looked at plant cork cells, which were relatively visible, and noticed them being in those shapes that kind of look like a cell, right? Circular catacomb shapes. And that's where the name came from, cellula. Apparently, there's some catacombs where people are buried to kind of have that shape where they're all kind of fitting around each other's circular shape. That's where the idea of cells came from, because it looked like little rooms. But he looked at cork cells. And again, he doesn't really know anything about nucleuses and all that stuff. He knows more about that than they did at that time, even being non-majors. Then there was a guy named Leeuwenhoek, who had a simple microscope where he would collect pond samples, and there's little algae and little critters running around. He saw that in a speck of water with a magnifying glass. And he started to describe them and see what a cell could look like amoebas and single cell stuff. He was able to start seeing that kind of stuff. So this is the first recorded information about microorganisms. And then there was another person around 1838. So this is still, think about this. This is still before the Revolutionary War and things that we think about in US history. And then not too far before the Civil War, they start to get an idea about cells. So we're talking 1838, 1839. So about 30 years before, more or less, the Civil War, right? In US history again. And there was a guy named Schwann and Sutherland that started to provide some of the ideas of cell theory. And that's what we're going to get into in a moment. This is the cell theory. This one's really important. It's the first concept is that all organisms, and I've already mentioned this a moment ago, all organisms on Earth that we know of that's living is composed of cells. That's one of the reasons why we don't say viruses are alive, because viruses are not cells. They're cell parasites. They need a cell to replicate, so they're lifelike. But technically, viruses are not living, for instance. All living organisms are composed of cells. Some organisms are single-celled, like bacteria or amoebas, a type of protist. Cells are the smallest living things on Earth. And then cells arise from other cells. So that kind of begs the question, how did the first cells ever show up? And we'll get into some ideas about that in another lecture. But for the most part, as best we know, cells come from other cells, not from non-living rock or something like that. And again, and I will make these notes available. I'll send you a recording so you can watch this also. So don't worry, we don't get every single detail. Does that sound good to everybody? All right, so when we visualize a cell, how big are they and how small are they? Well, this is kind of giving you an idea of your ability to see, right? So things like trees are around 100 meters, elephants 10 meters, humans 1 meter. All this is visible to the human eye. Hen eggs and when you're just talking about a chicken egg that hasn't really divided, that's a cell. You can see that with your naked eye. You can start to see the even small cells or big cells. I guess you should put it like single cell paramecium. It's a type of protist in the kingdom protist, or a human egg. You can sort of visualize that with the human eye actually. But it's right at the cusp. And then most of the cells are smaller than that, and you can't see without a microscope. And so things like human red blood cells, you need to have a microscope to actually visualize and see that cell. Bacteria, you need a light microscope. And I'm sure you've seen you used a light microscope in a biology class of some sort, even in high school, you probably came across one. Then we start to talk about things like viruses, which aren't living, they're lifelike. And at that point, you need an electron microscope to see it. An electron microscope shoots a beam of electrons and provides the image. And then we have things like proteins and amino acids that you can still make out with an electron microscope, but a light microscope will no longer cut it. You can't use a light microscope to see those things. Some of that has to do with how lenses work and the diffusion of light and stuff like that, but we cannot make out viruses, ribosomes. Does anybody remember what a ribosome does? I haven't got to that yet. Protein. Yeah, it's important for making proteins. Excellent. And then we can see small molecules like amino acid chains. But we really can't make out atoms. That's too small for an electron microscope. Okay. Here's Van Leeuwenhoek, and he's using his simple microscope back in the 1600s. And this is basically what it looked like. Here you can see it's just a staging thing with a little pin that you can put a sample of water on with a magnifying glass and then put it up to a light and then see a variety of critters or anything that was small that's floating around in some type of medium like water. So this is the first recording of microorganisms that we know of. Over here is how a light microscope works. You probably will have experienced this in lab or at another lab. And of course, it has various magnifying glasses or objective lenses. You put a specimen on the stage, you should be working with the lowest magnification, moving the big dials first, get it focused. You can switch over to higher magnification and start using the small dial. Hopefully they taught you that somewhere. And then you move to the next bigger. Hopefully you learned a little bit about the practical use of microscope. Now remember, once we start talking about really small stuff, we're going to need an electron microscope. So things like viruses, ribosomes, and things like that. Here is an example of an electron microscope. We actually have an electron microscope over in Kearns. In the chemistry department, we have a scanning electron microscope. We used to have both in this department. Those microscopes aged out, but we do have a state-of-the-art scanning electron microscope over in the chemistry department that we can utilize. Here is examples of what different microscopic images look like. You can see with the light microscope, we can pick out like this cell here. And you can see a lot of the details. You might see a nucleus. This is a scanning electron microscope. It scans the outside of the cell or an image of some thing you want to look at. So they take good pictures of what's outside. Then we have transmission electron microscope. We call it TEM. And you can see the insides of organelles really well. And you can even pick out ribosomes and stuff like that. And so those are examples of the different types of magnification and things that you might see. This can all zoom in a lot closer as well, but this still gives you an idea of the overall image. I don't know, I think we talked about this already, but we have 3 domains when it comes to, I don't know if Dr. Hahn mentioned this, but we got domains, then it goes kingdom, phylum, class, order, family, genus, species. Anyway, we're in the kingdom animals, for instance. Our cells are eukaryotes. We'll get into all that in a minute. So that's a domain, that's higher than the kingdom. So we have 3 domains. We have a domain of bacteria. And these include things like what's one of the most famous bacteria we all know? E. coli. So that would be E. coli, an example of E. coli, or a bacteria that causes strep throat. Those are all examples of everyday common bacteria that we think about. But there's also, okay, and so what do we see in the cell, just to make sure you understand? Inside the cell, it's very simple. We're just gonna see coils of DNA and lots of ribosomes. And these cells have a cell membrane that's flexible, and then they have a hard outer shell that we call a cell wall. Okay. You can find, I'm sure there's bacteria all over this tabletop, for instance, in the air. We also have another group of unique bacteria that's so unique that we actually put it in its own domain, archaea. The cell wall is dramatically different. And these are bacteria that we call extremophiles. They can live in extreme environments, super hot, near the ocean vents. And when you think about the hot springs and the Yellowstone Park, there'll be bacteria that can only live in those hot springs and sulfur. Inside, they look kind of the same. They got DNA running around and ribosomes, but their cell wall is dramatically different. So the idea is they diverged on their own evolutionary directions back when we had the first cell. We'll get into more of that in another class. Then we have cells that make up us. We call them eukarya, so that we're in the domain eukarya, which means true cells. The EU is true cells, eukaryotes. So we're in this domain. We're in the kingdom animal. What other organisms are in this domain? Plants, fungi, protists. Protists are things like algae and amoebas and stuff like that. So we got most of that. I think that's all 4. That's the 4, right? So there's 6 kingdoms, at least. Animals, plants, fungi, protists, bacteria, and archaebacteria. So that means you're more closely related to plants than you are to E. coli. Because plants have a true nucleus, just like you do. So these are all the organelles that you could find in a plant cell, for instance. you can find a nucleus. Inside it, you see a nucleolus. What does the nucleus do, by the way? Just in general. Yeah, it holds the DNA where all that kind of stuff happens. Inside the nucleolus, that's where the ribosomes are made. Outside of the nucleus, you'll see this other bunch of membranes with little ribosomes attached to it. That's known as the rough endoplasmic reticulum. Don't worry, I'll get all these names out to you later and more stuff. There's ribosomes. What does that mean? Well, that organelle is important for making protein. Good. And then we have what we call smooth endoplasmic reticulum. They call it smooth because there's no ribosomes attached to it. Does anybody know what that does? Well, that's important for lipid synthesis. Remember, lipids, one of the macromolecules. Then we have a Golgi apparatus over here. Its job briefly is to package proteins. Then we have mitochondria. So plants even have mitochondria. You might not have realized that. Does anybody remember what mitochondria does?
Powerhouse of the cell.
Yeah, right. And it makes ATP. It takes oxygen, breaks down carbohydrates into carbon dioxide. I'll get back to that in a moment. And then we have chloroplast, and it has chlorophyll in it. And what does that do? Photosynthesis. It takes in carbon dioxide, makes it into sugars, a macromolecule, and then when some herbivore comes around and eats that plant, that nutrients from the sun and the photosynthesis gets put back into the ecosystem. And so when we eat something, our mitochondria breaks down that sugars, those fats, into ATP energy in the mitochondria. We call that cellular respiration. It makes ATP, and this organelle, the mitochondria, is responsible for why we have to breathe in oxygen and why we breathe out carbon dioxide. That organelle in all these cells is responsible for it. But here's something kind of interesting. A lot of people know that basic story. We breathe out carbon dioxide. Plants suck in the carbon dioxide, convert it into food through photosynthesis. The byproduct is oxygen. That's the basic story everybody knows about, right? Since we're kids, we've heard about that in some class. But one thing you might not realize is plants also have mitochondria, and occasionally, and they will use a little bit of oxygen and release a little bit of CO2. Just not the same huge proportions. So that might not, something you might not realize is plants do breathe a little bit of oxygen too, particularly at night. Because remember, photosynthesis occurs when the sun's out. And when the sun isn't out, they'll break down that sugar for energy and release a little CO2 themselves. The interesting thing is when it's around twilight and there's not that much sun, they will do both at the same time. So they'll release a little bit of carbon dioxide from the mitochondria. That carbon dioxide will go to the chloroplast and it's kind of be internally circulating. But when photosynthesis is really occurring when the sun's out, then that's when you get a lot more oxygen released and carbon sucked up. And you can even see, you've seen models, if you see models of climate change, once it hits the growing season in North America, you'll see that carbon dioxide levels really drop when all these plants are growing. Okay. Do animal cells have a cell wall? Good. No. Do plants have a cell membrane?
Yes.
And well, you guys are excellent. Okay, so we already talked a little bit about the nucleus. We said that that's where the DNA is. And inside the nucleus, we also have a nucleolus. And do you remember what the nucleolus? My voice is changing. Must be going through puberty. Does anybody remember what the nucleolus does? Excellent, good job. And then when you got this nuclear membrane, it's like the cell membrane or the plasma membrane. It's a lot like it. Do you remember what the cell membrane or the nuclear membrane is made up? What kind of fats are they made up of? There's a special name for the lipids, these lipids here that make up the plasma membrane of a nucleus or a cell membrane. What is the name of? And there was a macromolecule, it should have been in your notes. That maybe you did or will be doing, I don't know. What was the name of the lipids that make up cell membranes? Does anybody remember phospholipids? So phospholipids are important for making cell membranes. Did Dr. Hum have a chance to talk to you about those things in class? Yes or no? Anybody remember? I may provide you a little online lecture to see that. Or you can just read her notes, but if you watch the lecture, you can see how phospholipids are important. If you want to watch, I'll give you that as an option. Does that sound good for y'all? If you want to, but I would recommend it. Or at least watching it up, and then the nice thing about YouTube videos is where I post these, you can fast forward and do whatever you need to, take notes easily. Anyway, and don't forget to like and subscribe to my channel. Anyhow, what else can I say about the nucleus? Oh, it does have pores. See, these are made up of proteins that function like doors inside this lipid bilayer that allow messenger RNA to exit and ribosomes and all that kind of stuff. We'll probably talk more about the cell membrane in a future class, depending, in the next class or something like that. This is DNA, or represents DNA. Obviously, it's a drawing. This is a double helix, and again, I don't know if Dr. Hum had a chance to talk about this with you. This may be something, I may just go ahead and provide you a lecture to watch that, because I talked about all this stuff. Or you can just copy your notes or watch a YouTube video or two to get the gist of it. Anyway, this is DNA, and then it gets coiled up around protein. So remember, the DNA is just floating around the nucleus most of the time. You don't see chromosomes normally when you look at a cell under a microscope. The only time you see chromosomes is when the cell's about to divide. And what do we call that? And what's the process when the chromosomes are lining up and mitosis? Go back home and slap your high school science teacher if you really don't know that or never heard that term. You guys are kidding, right? You've heard of mitosis.
Oh.
I know you've heard it, right? I actually taught them in high school, believe it or not, briefly during COVID. I'm doing both places. When the cell's about to divide, we'll get into that in this class anyway, and don't slap your high school teacher. You might not have been paying attention, right? Everybody's heard of mitosis, right? Meiosis. What's meiosis?
Something I never understood.
Well, I'll make it simple for a second for you. Both of them are cell division. Mitosis occurs in cell division of your skin, for instance, your muscles, your body. We call them somatic cells. Meiosis is cell division of sex cells, like sperm and eggs. And so what happens in that case, half of the chromosomes are removed. Because remember, we don't want sperm with a full set of chromosomes. Or eggs with full sets of chromosomes, we only want half. And so when the sperm and egg come back together, we have another whole person. So meiosis is about dividing those chromosomes in half. But it's still cell division of sex cells. Does that help a little bit? In a nutshell? I mean, there's more to the details, but now you realize that meiosis happens in our gonads, or ovaries, or testicles. When sperm's being made, or eggs are being made. We'll try to add that to the class at some point. Anyway, this is showing you how chromosomes are formed, and that happens during these cell divisions. All right, mitochondria, the cliche that everybody knows is the powerhouse of the cell, which is pretty simplistic for what it, but ultimately it makes a fuel source called ATP, androgen triphosphate. I'll show you a picture of that at some point too. I'll tell you a few things that are interesting about mitochondria. It has a double membrane. Remember the phospholipid bilayer? It has its own DNA inside it. So it functions almost like a bacteria living inside your cells. Okay. Here's something else interesting. The mitochondria in your body and your cells, mostly, in most cases, came from the mother's lineage. So that means all the mitochondria in your body came from your mom's lineage and her mom's lineage. That's one thing. Why is that? Well, when we have the egg, we have mitochondria and all that other stuff in it, all the other organelles. When the sperm comes swimming, finding that egg, the only thing that really penetrates is the sperm will come, the nucleosome of it, open up and shoot in the chromosomes. The mitochondria is all in the tail. So that doesn't enter into the egg. So that's why the organelles of your body came from your mother's lineage, or at least the mitochondria. Here's something else interesting that made people start to wonder, because remember, what about the simplest cells? They're like bacteria, right? The simplest cells are like bacteria. Well, no. What I mean by that is those domains like bacteria and archaebacteria, they're just membranes with DNA in it. And these mitochondria act like bacteria in your cells. They'll divide on their own inside your cell. So what they think happened in the early Earth's history is that a big cell came along and ate, swallowed one of these bacteria that ultimately became a mitochondria. Instead of digesting it, that cell lived inside it, provided energy to the big cell that's housing it, and they formed what they call a symbiotic relationship. We'll get into that a little bit more detail, but it's called the endosymbiotic theory. And why do they think that a little bit? Because again, this runs around inside cells, dividing and things like that. It also has a double membrane, and it has its own DNA. Because otherwise you just thought DNA was probably just in the nucleus. Now you realize DNA is also in the mitochondria. They also think the same thing for chloroplast, that a big cell engulfed a bacteria that could photosynthesize, lived inside that cell doing photosynthesis and providing nutrients to the big cell that engulfed it. And again, forming a symbiotic relationship, a lot like the mitochondria. Chloroplast also has its own DNA. Circular DNA. Remember, bacteria have a circular DNA. So that's why they think the chloroplast functions like a bacteria inside plant cells anyway. And plant cells have both mitochondria and chloroplasts. If it wasn't for chloroplasts, life wouldn't exist on Earth. At least on our life we care about. There may have been some bacteria that eat some sulfur or something like that. But this is why the atmosphere has oxygen in it in high amounts. It's thanks to chloroplast. Inside chloroplast, we have a substance we call chlorophyll. It absorbs light and it hooks up CO2s into chains that ultimately make foods and sugars that the whole ecosystem is based on. You know the story, herbivores then eat the plants. Omnivores and carnivores eat plants and onward animals, and then carnivores focus on it. All right. All right, here's a little description about plastids, which are related to chloroplast and some stuff we were talking about. So plastids are double membrane organelles found in plants and algae. And there's different examples of them. And so they're going to do things like storing food and manufacturing food. They're also going to have pigments for for photosynthesis, like you see in chloroplasts. And there's different types. Here's plastids you would see under a cell, for instance. And there's actually 3 different types. So we have things like chromoplasts, leucoplasts, and chloroplasts. And apparently each of these have different types of functions. Of course, we always think of chloroplasts, or at least I do, when it comes to photosynthesis. So these are have these membranes that are found in a double membrane bound chloroplast. So that's where you're going to find chloroplasts. And again, that's what's going to allow for photosynthesis. Yeah, 1 second. All right, so that's the gist of those, and I hope you appreciated this presentation. Take care.